Today's guests are Janet Whiteside and Mark Muller. We join their conversation in the faculty lounge.
So what happens when when you hear low millibar readings on hurricanes? This one went down to I think 902 millibars, which is really unbelievably low. I think the record anywhere on the planet is like 880 or something. What happens in those low pressure situations, according to what I've read, is that you get a dome of water that just gets drawn up. And of course that's a storm surge that winds up coming inland. And I mean, it's just really hard to recover on a deal like that.
Yeah, we lived in Panama City for a while, and we were on a bayou. And coming from up north, I'm from Illinois, had no idea, even though we were inland and on a bayou, I had no idea that really the water was all going to come in. You know, when the surge came, it's going to get locked in the bayous, and we were going under. So I learned what floodplain A meant.
Make sure your flood insurance is paid up.
Yeah,
I hear you. Yeah.
Well, can you tell me some about your research? Because I'm personally interested.
Sure. Well, let me tell you, first of all, I was the director of experimental therapeutics at Ohio State in the Comprehensive Cancer Center.
Okay.
And I had a number of, I was co-director with a physician, and I had a number of clinical oncologists that worked under me. We did clinical trials, and I've been involved in a number of clinical trials, including some that you mentioned that your husband was treated. And so I see the work as a basic scientist, which I'm trained as a researcher that does fundamental mechanisms of gene control, that kind of stuff. And then I see the other side of it, which is sort of going from the bench to the bedside, what the clinicians see, and it really shook me up. I mean, just to give you an example of where I'm going with this, I was in a clinical trial a few years ago with a collaborator, a guy who's a hematologist actually, and one of the targets we work on is a protein called DNA topoisomerase, which is a DNA damaging agent. I can tell you more about that, but we discovered a very potent inhibitor of this enzyme, which then went into clinical trials.
Okay.
And the drug, it's a cytotoxic, really nasty drug. And when you do a phase 1 trial, it's to determine the maximum tolerated dose of the agent. And in this particular case, we knew that this would kill tumor cells, but we weren't sure about collateral damage. And we had, of course, end-stage patients. These are patients that are terminal and all hope is abandoned. And these patients are put on the trial, and we actually found that about 80% of the patients died immediately as a result of therapy. And this is nasty stuff. But on the flip side, around 18 or 20% in the study went into remission and actually were cured by definition of a 5-year survival. So we had basically 1 patient out of 5 that was going to recover from therapy, and everybody else is going to succumb and directly as a result of the drug. So these cancer therapies are poison darts, and you don't know. It's like you have a guy treading water. You don't know if he's going to get an anchor or a life preserver. And they have no idea in many cases with these compounds. Now there's a plethora of anti-cancer drugs out there. Who's to say what's going to work? So there's got to be what I'm saying is there's got to be a more rational approach to the problem, because you've got to know more about the disease, you've got to know more about the biological markers, you've got to know more about the targets. So what I'm trying to develop, what I'm interested in, and it's kind of in my field, it's a truism. Everybody goes, yeah, mom and apple pie, right? Everybody wants this. But I always feel like I need to say it. We need a predictive model for therapy. You need to be able to say to patients, look, I'm going to give you this drug and you will respond. There may be some side effects, but the tumor is going to go into remission. And that's what's missing. Right now it's precedent and statistics, and this cohort responded in this way, so we're going to give you that drug. Well, we're individuals. We're not numbers. So even though statistics say that the majority of the people respond to this drug, if you don't respond, it doesn't mean anything.
That's right. Yeah.
So the way cancer research is done these days, an important part of anti-cancer drug development is you take a mechanism and you expand on the mechanism, and then you find out where the Achilles heel is in the process. And then you try to develop a drug that hits that step. So it's very tractable, it's very well defined. And my feeling is we should expand that to individual patients. We should take patients and we should, the first thing you do in cancer, as you know, is surgery. If it's a solid tumors don't have a good prognosis, by the way. I mean, that's a bad deal. And there's very, very few agents that work well with solid tumors.
Now, what do you mean by a solid tumor?
A solid tissue, as opposed to leukemia or the soluble, what we call soluble tumors.
And that does not have as good of a
There's
rate?
fewer therapies available for solid tumors.
But I thought it was better because you can actually remove it, surgically remove it.
Right, but the problem is they spin out a lot of cells. The cells are mobile. They have a tendency to break free and establish
Okay, now I've always heard, like some people have told us, like if you have a solid tumor and you take it out, then you've increased the chances that it's going to spread. Is that a fallacy?
I think that's a case-by-case deal.
Okay.
Every one is different.
Okay.
You have, highly metastatic disease and the cells that are very aggressive, undifferentiated cells that can break out and make microtumors. My mother actually died of lung cancer a year ago. And there was that was a situation where the primary tumor was in the lung. She wasn't a smoker, but non-smokers die of lung cancer all the time.
Yes.
And the primary was in the lung, and they picked it up on an X-ray. And this is what really irritates me: that the doc, her GP, picked up this very small tumor mass on a routine X-ray and missed it.
Missed it.
He missed it. I should say she. And I went back and I pulled the films and I could see it. And by the time they diagnosed it, it was the size of a baseball. And when tumors get that big, the number of microtumors, microcolonies that are spun off becomes unwieldy, and it just populated her lungs. There was no recovery. So I guess my bias is we need to know more about individuals. I was reading recently, there's a company, I can't remember, I think it seems like they're in Madison, Wisconsin, that's got a patent on a genetic test. And if you have a particular mutation, then when you treat with this drug, and I think the drug is a topoisomerase poison, it's related to camptothecin and irinotecan. If the patients have this particular genetic predisposition, they will not respond to the drug.
This particular
drug. So it's a negative indicator. And in another case, in other cases, it should be possible to develop strategies where you profile a tumor, and then you can say they will respond.
Yes, and
there will be more collateral damage if they have this particular pathway or this particular sort of biochemical profile.
Right, and they're doing some of that, aren't they? Yeah.
That's what a lot of tumor profiling is all about.
Okay.
With the new array technology and whatnot. So we're really interested in this aspect of science, and I'm really interested in translational research, which means going from the laboratory directly to the bedside. So
I like that.
So we
because I've been at the bedside.
Yeah, we want to be able to connect what we do in the laboratory with what's happening in the clinics. And one of the things I bring to the table with clinical trials is we're really good at doing biological correlates. You know, there's only so much you can do with blood chemistry and what I call feet up, feet down. You know, the patient responded or didn't respond. You need to be more insightful. You've got to be able to see a specific endpoint in a clinical trial, and that's what we've been really good at, is developing sort of in vivo, that means inside the body, tests of response to therapy. The idea, of course, is that once you develop a profile of patients and tumors, you should start to see associations where, you know, if this patient is expressing this gene and is showing these symptoms and you have a tumor of this sort, here's a drug of choice.
Right.
This will work. This drug over here is going to kill them.
Okay.
And that's the truth. Some of these drugs aren't neutral. They're cytotoxic agents, and it's either a home run or it's going to put you in the dugout.
Okay, meaning at the cellular level that this drug is going to actually kill the cell, or it will kill healthy cells?
Healthy cells, exactly.
Okay.
So, for example, let me tell you about the work we did on this agent, topoisomerase. This is a DNA binding protein that's present in all our cells and all our bodies.
Okay.
And it's required for anything that DNA does. DNA,
Okay.
of course, being the genetic material. So we know from studies from simple organisms, if you eliminate this gene for topoisomerase, it's a big problem. The cells don't like it. So here it's required for cellular function. Now, the enzyme does a very simple thing to DNA. It's real easy to understand.
Okay.
It makes a single strand nick.
Okay.
But DNA is it's
Yeah.
a double helix. So
Right.
here's DNA right here on
Yeah,
my tie.
I like that
right?
tie.
So here's the DNA molecule.
Yeah.
So it actually, what it does, if this is a double helix, it makes a single nick in one strand.
Okay.
And the reason that's important is in order to read the information content in DNA, you have to separate the strands.
Okay.
And that creates a bubble in the DNA which
Okay.
propagates. To adjust DNA structure, you need to make nicks in the DNA.
Okay.
So here you have an enzyme that cuts DNA.
Okay.
That's inherently dangerous to a cell because if it cuts and doesn't reseal it, then you've damaged
Right.
the genome.
Got it.
But the topoisomerase has evolved in an interesting way. When it makes a nick in DNA, it immediately reseals it.
Oh, that's
So it's a
good.
coupled nicking and resealing to protect the genome. What these drugs do that we've discovered is when there's a nick in DNA, the drug insinuates itself stereochemically in the nick, and it prevents the resealing. So now the drug turns a beneficial enzyme into a DNA damaging agent. And that because tumor cells have typically slightly higher levels of the target in this topoisomerase, then the tumor cells acquire more DNA damage than surrounding cells. So
I
you have
see,
a
okay.
quantitative, or I should say a qualitative approach
Right.
to knocking off tumor cells, right? So that's the model, okay? So now let's imagine, so now the target has got to be present. The target is the topoisomerase, it has to be present in order to see an effect. Well, it turns out that some tumor cells have very little topoisomerase. If there's very little topoisomerase, there's no reason to give them the drug.
No.
And that's a simple
Okay.
test you can do
Right.
that all clinicians should be able to say,
Okay.
what's the level, and we actually have a paper that we're in the process of writing up that describes this with myeloma, with studies we did with myeloma. And basically we found that some of the multiple myeloma lines we looked at have virtually no topoisomerase 1 or barely detectable. These cells are completely resistant to this drug.
Right. Yeah.
And if you have a patient and you give them this drug, they're not going to like it very much. And the tumor it's not going to do anything to the tumor.
Right. Yeah.
So it's a double-edged sword. When it works great. If it's not there, you better stay away from that
Right.
compound.
Yeah.
So I've seen the side of the field where these agents are invasive and cause a lot of collateral damage and can be, really, really bad things. In some cases, they work beautifully. But you've got to know that ahead of time. You can't just throw these things out
Yeah.
and hope that the patient lands on his feet.
That's right.
That's the
Yeah.
rub. Now, a second project in the lab, we've developed an interest in telomerase, which is another target, another cancer target. And telomerase is more quantitatively elevated in tumor cells. It appears that tumor cells, 80 to 90% of tumor cells have telomerase, and virtually 100% of somatic cells, the rest of the cells in our body, don't.
Oh, okay.
And what's interesting about this concept is it's a relationship between cancer, cell growth, and aging. And it's an interesting story in that normal cells in our bodies divide about 50 times, and then they stop.
Okay.
We call that the Hayflick Limit, named after the guy that discovered it.
Okay.
But basically, there's a mechanism inherent in every cell in our body that says you can divide precisely 50 times, and then you will stop dividing. And this was discovered by taking cells and cultivating them in vitro, in test tube, and showing that after a certain number of population doublings, the population stops. And no matter what you do, you can give them new serum, you can give them growth factors, you can do whatever you want, but they stop. And so if you extrapolate that to humans, if I were to take, I told you about my father had his 90th birthday. If I were to take fibroblasts from my father, do what's called a skin punch, no, he wouldn't like it very much, but I could probably talk him into it. Take a little bit of tissue and put it in culture. His fibroblasts would probably divide 2 or 3 times.
Okay.
If I did it to a neonate, a 2-year-old, you'd probably get 48 population doublings from that individual's fibroblasts. So it translates to the patient, to the person. Biological relevance is there. So what's the basis of this? It turns out that it has to do with chromosome ends, believe it or not. The physical ends of the chromosomes are called telomeres. And they have a peculiar property in that this DNA doesn't encode anything. It doesn't make anything. It's just there. And it has a bizarre repetitive nature. So it's like it's repeating DNA that goes out a long way in normal cells. What happens is that when the cells divide, there's an inability to replicate the physical ends of the chromosomes. And as a result of that, every time a cell divides, there's a way of ticketing the number of replication rounds. So there's a ticketing agent that meters how many replication events have occurred, because every time the cell divides, a little piece of the end of each chromosome gets clipped off.
Ah, okay.
So it's what you replicate once, you clip this.
Yeah.
Replicate one more time, it gets clipped again. This clipping occurs until you erode the DNA into real DNA, which codes things, and the cells just die. They go into what's called programmed cell death. Now, tumor cells, and actually you've heard of stem cells probably, these primordial precursor cells, immature cells, and embryonic cells, they have an enzyme called telomerase that prevents the erosion of telomeres.
Oh.
So if the telomerase is present and the cells divide, they're immortal because the ends of the chromosomes don't erode.
Yeah.
So this process propagates, and because tumor cells are telomerase positive, they don't experience this ticketing process, and they continue to replicate.
Got it.
All right, so the question is, normal somatic cells have no telomerase. And that's been the real question. Why?
Yeah.
How is that?
Yeah.
So, and even more importantly, is there a way we can eliminate telomerase from tumor cells? Because then we can treat tumors. And my prediction is that if you can eliminate telomerase in a biological, subtle way, then you can prevent or avoid collateral damage, because all the other cells in our bodies don't have telomerase.
Right.
So it's
they'd be okay.
so this has gotten me interested in this whole area in the last few years. And one of my associates, a former postdoctoral fellow, he's from Korea, and he went back to Korea and he set up a lab a few years ago. And he's really a very gifted scientist. And we've been collaborating on this project for several years. And we had a really important finding that was published in April, and in a good journal in our field. And actually the journal, not me or not my collaborator, but the journal issued a press release on the findings because they thought it was pretty important.
Right.
What we've discovered a mechanism that telomerase is eliminated. And that was a very nice piece of work that involved a team of about 6 or 8. There were 2 labs in Korea and Seoul, and then my lab at Ohio State, and then we continued the work after I moved down here to University of Central Florida. What we discovered was that telomerase itself is actually kind of unstable, and it has to be continuously made in order to be retained inside the tumor cell. What we discovered was that there's another factor that modifies the telomerase, and when that telomerase enzyme gets modified, it's immediately degraded. It's put in the trash can. And we cloned that gene that does that process. So the gene is called macron-1, and essentially what happens is the macron-1 encodes what's called a ubiquitin ligase, and I don't want to get into the scientific details, but basically it's a step that adds a moiety called ubiquitin directly to the telomerase, and this marks it for destruction by turnover processes inside the cell. And what's really exciting about it is when we take the gene for macron and we put it in tumor cells, we eliminated the telomerase and they stopped dividing.
Right.
But which is a really cool experiment.
Yeah.
And an important finding. But there's good news and bad news. The good news is the cells stop dividing. The bad news is it takes a long time, like 10 population doublings, before it happens. In other words, it's not something that's immediate right now.
Right.
You put macron in, it takes 10 days, 14 days of growth of the tumor before the cells start losing their oncogenic potential. Now, that's a little bit of a negative, I realize, because it's not instantaneous, but it might actually be a good thing because it's subtle. It's not going to go in there and wipe out all the tumor cells overnight. It's going to be a gradual process, which I think might even work better.
Sure.
And give and have less of this collateral damage.
Now, would this work on any type of malignant tumor?
That's a really good question, and that's what we just discovered this, so we're desperately trying to find out the mechanism in all tumors. So, for example, one of the things we're doing, we have to make antibodies against this protein, and that's ongoing. So once we have antibodies against the protein, we can track where it's at in different tumor lines. We're getting the National Cancer Institute has a bank of about 60 different tumor lines, cell lines, that we're going to bring into the lab, and we're going to screen, and we're going to find out in what kind. Is it in brain? Is it liver? Is it colon? Is it leukemia? Is it myeloma? What have you? So that'll give us sort of descriptive measure of where the enzyme is. And the really interesting thing about what as a molecular biologist, I mean, I look at this stuff and it just amazes me because the gene we found, it turns out to be, we pulled it out of the Human Genome Database. So you know about the Human Genome Project, of course. And that's given us a resource. When you have a little bit of information about a gene, you can go into the public database for the human genome, and you can, in silico, you can pull that gene out. So life is nice. That was a huge investment in biomedical science in our field. And when we pulled the gene out and we analyzed it, we realized something. It's a really ancient gene. It goes back, even lower life forms have this gene, and it's virtually unmodified from lower, like yeast have it, and it's a primitive unicellular organism. They have this thing, and it hasn't evolved any. Now, why is that? Well, it turns out it's pretty important. genes that are critical don't change, because if you make them during evolution, if you
Right.
make a mutation in that gene, the organism dies.
Right, yeah.
It's negative. It's Darwinian, right?
Right, yeah.
So if you eliminate these things, it has negative survival value. So this gene has survived millions, if not millions and millions of years.
Right.
I don't know exactly what the breakout is, but it goes way back. And so here's a gene that's a founder gene for actually a gene family. And when we started looking in the genome, we found there are actually a large number, maybe as many as 10 different family members for this gene. So we've opened up a whole bag of pathways that we think
incredible.
impact on stability, regulation of proteins that are important in gene control, perhaps. And what we'd like to do is to develop sort of genetic inroads where we can maneuver the gene and put it inside cells that need to eliminate telomerase.
Right.
That's a tall order, and it's going to take a number of years. But it's a new model, and it's exciting. And so we're trying to push this. We're probably about 5 years away from having a real drug target, where we can say, here's a drug that's going to work, and because of the mechanism we've uncovered. It's good.